All right, good evening, folks. Thank you all so much for joining us. Welcome to the Starkville Planetarium. My name is Eric Johnson. I'm the director here, and it is my pleasure to host the James B. Kaler Science Lecture Series. During this series, we invite people who do research in the world of science or do lots of cool things in relation to science and share what their passions are with the public. The series was named after Jim Kaler, who was a professor and later a professor emeritus over at the University of Illinois. He was specialized in the field of astronomy, and he gave lectures here every year from roughly 1988 till 2018, 30 years of wonderful lectures. So after he had to stop doing that, we renamed the series in his honor. Anyways, for the last several years, we've also been able to showcase the summer research performed by community college students, and that's what tonight's talk is actually going to be all about. All right, we have a new program that the college is associated with. You see it up there on the screen. It's called MICRO-CCS, Microbial Interactions Create Research Opportunities for Community College Students. Yes, and we have the principal investigators here, the faculty sponsors of these student researchers. Our speakers that we will have this evening are Ray Elementi and I believe Zwawi. Zwawi? Okay, I wasn't sure if I pronounced that well enough, and I appreciate any corrections you have to make. I should have asked beforehand. I'm glad this is on tape. All right. Anywho, we are looking forward to seeing what they have to say, and we'll get also a short introduction from Dr. Chelsea Lloyd. She's a professor in microbiology here at Parkland College. She is one of those co-PIs of the MICRO-CCS program, and she's going to tell you also a little bit about the opportunities that college students have for seeing if these scientific fields would appeal to them in terms of that future research. All right, so who is our first speaker for this evening? It is Dr. Lloyd. So Dr. Lloyd, if she needs further introduction, she is an alumna of the University of Iowa, go Hawks, and she is also she earned her PhD was it in microbiology? Mm-hmm. Yes, at the University of Illinois. Soon afterwards, she came to Parkland as one of our professors in the Department of Natural Sciences. Did you want to speak with this?
Yeah, that's great.
Okay.
Tag me in.
Yes, and here is the remote.
Thank you.
Right, I will start to get those jack-o'-lanterns off because that would be distracting otherwise.
Oh, I think they're delightful. Hello, everyone. So I'm going to talk a little bit first before I introduce our speakers about what on earth they did this past summer and what exactly an REU is. This is what we call a research experience for undergraduates. And this is my contact information as well. So if anyone has any follow-up questions, you're welcome to reach out to me. So if you're a STEM major, is anybody here a STEM major? Okay. You know how hard this is, right? STEM is very difficult. And so research has shown that research experiences that undergraduates do actually improves your outcomes in STEM. And part of that has to do with confidence in what you're doing, getting to actually experience and participate in the scientific endeavor. It also helps connect what you're learning in your classes to real-world applications, and it can also help you feel a sense of belonging in a field that may otherwise be very othering. It's sometimes hard to fit into science. And so these experiences you can get in a variety of different ways. There are several unpaid ways you can get research experience. So you may do volunteering or shadowing in a laboratory. You can do research for credit. There are designated courses for research. You might even be taking a class that has a research component built into it. So my microbiology class has research built into it as well. But if you want to be paid, there are ways to do that as well. You could get a research assistantship or a research internship, but those are sometimes difficult to get or difficult to find because they're often unlisted. You kind of have to network and know people in those settings already, and so the equity isn't really there. And so there are federal programs that will pay you to participate in research, and that's what REUs are meant to do. So federally funded undergraduate research programs pay you a stipend, so you get paid to do this work. You're often also housed, and your transportation is covered as well. So for example, our program, we had 2 students from California. We flew them in, we housed them over the summer, we gave them food allowance, and we also paid them $7,000 for 10 weeks. So they're really great opportunities, but they are very competitive. So you do have to apply, and there are often hundreds of applicants for very few spots. So I encourage you, if you're interested in applying, to apply to many opportunities. And increasingly, these opportunities are looking for community college students. So is anybody a community college student in the audience? Yes. Okay. So you are very valuable. We really want you to participate in research because we're trying to broaden participation. And we want your lived experiences. We want your perspectives to contribute to science. And so there are more and more opportunities exclusively for community college students, such as ours, but the Department of Energy, NASA, the NIH are examples of programs that are also for community college students. But you're also welcome to apply to other opportunities for undergraduates in general. So this is a commitment. It is a full-time job. It is 40 plus hours a week. So that's something to consider. And these are over the summer, typically 6 to 10 weeks. And that may not be tenable for everybody. But they're also responsibilities after the REU. Because these are federally funded, we need to report back to our funding agencies and let them know what you've been doing years after you've left the REU. So you do have to stay in touch with us years later so we can report back to the taxpayers what we've been spending their money on and make sure that these initiatives can keep being funded. So some of the benefits are discovery. So it is very cool to be the first person to discover something new. You're doing actual research that's pushing the boundary of knowledge. You're not just doing some canned laboratory experiment that we already have an answer to, which a lot of coursework does that. So you're actually discovering new things. And you get to discover new things about yourself. I think our speakers tonight are going to discuss some of the things they learned about themselves during this process. And it's important just to figure out what you like and what you don't like. If you don't like research, that's just as important to know as if you love it. And obviously, this is going to pave the way for future opportunities, and you may even really fall in love with your summer research lab and continue working there like these two both have. So this can open new doors for you even if you're not really into the thing you did your research on over the summer. So challenges, because this is full-time work, you cannot work another job or take summer classes when you do an REU. And so this may not be feasible for people. You may have a job that you may not be able to leave for 10 weeks over the summer. And so this type of opportunity may not be right for you at that time. We also have people who have family obligations or healthcare networks that they can't leave for 10 weeks. So we understand that as well. And so those might be things that might discourage you from applying. But there are some ways that you can have some flexibility built into this, and I'll show you an example of that later. You also have to adhere to the expectations of the lab. So that might mean coming in on weekends or coming in at weird hours of the night to take a time point. So that might be something that's challenging, as well as failure. There's a lot of failure in science. That's just kind of part of the business. And so dealing with failure might be a challenge for you. You might also have personality clashes with people in your laboratory. You might also have more practical issues like health insurance. Some programs pay for health insurance, others don't. So those are just some practical challenges, but I would say that the benefits definitely outweigh those challenges. So how do you find these opportunities? A couple of really great places to look are pathwaystoscience.org and the NSF National Science Foundation ETAP. This is the education and training application. And both of these websites are great because you can search by topic, by field of interest. You can search by geographic location if you're constrained, or if you're like, I want to get out of Illinois this summer and try Washington State, whatever, you can search by that. And you can also search for opportunities that are exclusive to community college students. That's something not everybody gets to do. And the nice thing about the ETAP is you can also apply at this website. So our REU is housed there, but you can apply for that one and many, many others all in one place. So it's meant to make it easier for you. Also, if you're interested in local REUs, you can Google UIUC REUs and you'll come up with this website, and that'll help you look for local opportunities if you can't travel over the summer. There are also international opportunities, I should mention, if you are interested in leaving the country over the summer. So our program is interested in microbial interactions, not just between microbes and humans, but microbes and plants, microbes and other microbes, even microbes and insects. And there are a lot of different disciplines intertwined in our program. There are engineering labs, there are people doing computer science informed microbiology. So there are a lot of interests that can overlap with our topic if you're interested in our program. Some benefits of this are the $7,000 summer stipend. I would say that's pretty competitive compared to some of the other programs, just saying. And one thing that makes ours unique as well is a research orientation boot camp that I'll talk more about. The nice thing also is we don't have a GPA minimum, and that's because we understand your lives may be complicated. You know, things happen. You might have had to drop a class, or you might have failed a class, and that's okay. We want to hear about what happened. You can turn that lemon into lemonade and talk about how you're resilient and you don't stop with failure, which is a great thing for a researcher to be able to do. So we don't have a GPA cutoff, and we don't expect you to have research experience. In fact, we prefer applicants that don't because we want to give you that opportunity. And we do recommend that you have some biology and chemistry under your belt, but we don't have any course requirements. So what is this boot camp? So we understand if you haven't done research, it might be really scary and terrifying to try to apply for this opportunity. You may have some imposter syndrome, thinking, oh, I could never do this. That's okay. We have all been there. And so we have a research boot camp for the first week of the program to get you acclimated to the lab space. So if you've never taken lab courses before, or if you have very little lab experience and you have no research experience, we're going to teach you some basic microbiology, chemistry, and molecular biology skills so that you're more comfortable and confident going into your actual research lab. We also help you get to know your cohort a little bit more. We'll do some social activities. We also have some opportunities for you to meet your research mentor ahead of time so you can talk about your project expectations and come up with a contract together. That way, when you step into your research lab, you'll be more confident and comfortable getting started. And then you'll spend 9 weeks in that research lab doing cutting-edge research at the University of Illinois. And then after the summer's over, you'll present your findings at a research symposium. So if you want to apply, our application will open December 1st and it'll close March 3rd. I would say if you're interested in REUs, start getting your materials together now because some of these REUs start closing their applications in January for the summer. So I know it seems like a long time from now, but start putting those application materials together. What you'll need for most REUs, including ours, are a resume. Work experience is really valuable. Food industry work experience is a really great thing to put down because you've worked with food safety, machines, people. You'll need unofficial transcripts is fine. You'll need one letter of recommendation from a faculty member or advisor, and then a list of your top 3 choices of labs that interest you. Now, the most important aspect of your application is the personal statement. This is where you get to explain if you have a W or an F on your transcript. We want to know who you are and what experiences you've had. Why are you specifically interested in our REU, and what do you want to get out of it? What are your goals? So your personal statement is your opportunity to tell us who you are beyond your transcripts. So if you have any questions, please feel free to reach out to me. If you're a Parkland student and you're on campus, you can also swing by my office. It's L132. I have snacks if you just want a snack. Candace knows. And this is our website. So we also have, in addition to the application, we also have information about how to make your application really competitive for this and other REUs. So please visit our website. We've also got a QR code here. I'll have flyers and my business card as well if you want to reach out. So with that, I'm going to introduce our speakers tonight. They were both REU participants this past summer, Ray Elementi and Adam Zwawi. Ray is the first one up. So I'm going to let him talk about what he did this summer.
So, just gonna start with a little bit about my path to starting to participate in this RU. I had a bit of a journey to get here, but I was a student at College of DuPage and had kind of encouraged other people to apply for RUs and was getting stuck in that imposter syndrome of not feeling like I was qualified. And then I was seeing the people I was recommending get these opportunities, and I took a chance, and I was really nervous and wasn't really sure where it was going to lead or if it was going to lead to anything. And it's been a really life-changing experience, and I'm so grateful for the opportunity. It actually has led me to now being a student at U of I. So I ended up being placed with the Stashevich Applied Food Safety Laboratory, and the mission of the lab in general is to apply engineering principles and kind of the concepts of engineering and the way we approach data and analyzing things and applying it to making our food safer. So as of right now, a large percentage of the foodborne illness outbreaks actually come from produce sources. And it's about 60%, as far as the data we have says, of E. coli 0157, which is the strain of major concern with E. coli. And that 60% of those cases comes from just vegetable row crops alone. And so we're trying to kind of find better ways to identify and stop risk of foodborne illness from getting to people. So one of the things we're looking at and prioritizing is pre-harvest soil testing. And the reason we're looking at soil specifically is because it's not only a reservoir for bacteria, potential pathogens to live, it's also how it gets to the surface of the produce that then goes all the way down the supply chain until it's eventually what you're picking up in a grocery store potentially. And so there's a bunch of different ways that bacteria can be introduced to soil, and then there's a bunch of different ways that it can reach the produce surface itself. So the methodology we're testing is adapted from the meat processing industry, where rather than just taking a swab and reaching in one drain and trying to get a good idea of what's out there by just swabbing individual things, the newer methodologies recommended are things like boot swabs or drag swabs, where you walk through an area and collect the sample that way. And so we're adapting this now to be used in produce agriculture, and specifically for testing soil. So the previous research showed that these swabs are effective in collecting relevant indicator bacteria, but now we're trying to kind of further optimize this process for scaling it up. It needs to be something that can be done on a large scale in a commercial agricultural field. Otherwise, what's the use? So trying to figure out, okay, is there one wetting agent that works better, or does it just matter that the swabs are wet before you walk over the soil? And specifically for what my work was to see how long we're able to collect relevant information that's able to be used to make decisions as to what we're going to do, how we're going to harvest, if we're not maybe going to take further steps to control or need to identify where the risk is for pathogens to be reaching the produce. So essentially what I did was I collected, for each walk, there's the two different drag samples, the two different booty samples, and then you also, the more traditional method of soil testing is to physically bend down and collect soil as you go. Obviously, bending down, collecting soil is a lot more onerous, samples heavier, it's harder to reliably scale that up, and it just hasn't been done. Whereas walking through a field and just collecting a sample that way, are you able to get good information? Are you able to actually take that and then process it within a reasonable timeline to make a decision as to how we're going to treat this produce? Specifically, when it comes to scaling this up for industry, in the lab we can collect these samples, process them, have the information within 24 hours. In a commercial farm, that's not realistic. They don't have a lab on site or people who can test it that fast. The samples are instead stored in refrigeration and sent to a lab to be tested and quantified. During that time, are we losing valuable information? That's why I took these samples, and what I did was put them in storage in the fridge and took 3 random sets of samples and tested them on the day they were collected, 1 day out, 2 days in storage, and then since we weren't really seeing a lot of change, we pushed it out to 7 days, which is the furthest it would be reasonable to really use that information to make a decision on a harvest and see what changes about the sample, if there's die-off in the number of relevant indicator organisms. So, kind of talking a little bit about indicator organisms, it's not actually super visible here, but unfortunately the color isn't super defined. But when we put the the soil samples that have been diluted and plated, we have a specific type of media that will identify if it's a coliform. It selects for coliforms, which are generally bacteria that grow in the intestinal system of animals, and so it's an indicator we use to see if there's been potential contamination in that area of soil. And so the purple is for coliforms, and then those are actually going to be bluer tinted. You can kind of see it better in this image where we streak plated for the larger project. But you can see there's like the blue little colonies, and then there's the purple. And the purple are just generic, are just coliforms, and it's not specific. And then the blue are actually identifying, okay, this is a generic type of E. coli. It's not necessarily saying that this is the strain of concern, the pathogenic E. coli, but it's an indicator that there is E. coli in the soil. And then we also, to kind of get a general sense of the microbial load of the samples, ran APC, which is just standard methods agar. If it's something that can grow on it, it will grow. And so these samples, you get some really interesting, cool kind of patterns that emerge in growth and resistance within the plates, but it gives you an idea for roughly how concentrated the microbial life is in that sample. So what we found from this was that the samples were surprisingly stable even up to the 7-day mark. You can see the 3 different indicator organisms, the general microbial load, the total coliforms, which indicate fecal contamination, and then specifically just generic E. coli. And what you see is that this actually shows it a little bit better. The counts on the samples after even 7 days of refrigerated storage are still quite consistent. And that tells us that it would be feasible to have these samples stored for a few days, tested, and still be able to provide relevant information that you can use to make decisions and action on. We did, however, see that by the 7-day mark, there was a significant difference between the counts you're getting at the 7-day mark, specifically compared to days 0, 1, and 2, but only for the APC, which is the just general microbial load, the standard methods agar, and the generic E. coli. We didn't see any significant difference, which is positive for us, in the total coliform counts, which is what we're primarily using as our indicator for fecal contamination, because of the fact that generic E. coli is in general very diffuse in the soil. It's not super concentrated. It's hard to you don't always find it. I actually intentionally took my samples during a day where it was raining, because that's considered a contamination event, because when it rains or when you're irrigating, that's what causes things to flow through the field, touch the produce surface, and also, let's say a bird pooped. Well, now that bird poop is spread, or the soil splashes as the raindrops hit, and then it hits the produce surface. So we can actually consider any irrigation a contamination event. And so that's when I took my samples then, specifically to try and make sure that if there was E. coli, that was our best chance of having the highest number to then really see how that was dying off in the samples. So what's going on now with this research is doing a lot of data analysis. I have a little Halloween relevant joke about normal and paranormal distributions. But we also, with the samples for the larger project, took some of the samples and processed them to do DNA analysis, and so we're working on the bioinformatics for that. We're working on collaborating with a soil-specific laboratory to give us a better understanding for how this research translates into different soil contexts, because we have tested in different types of fields. And we're writing a paper. Then, me personally, I'm working on this project, but I've also developed into starting to work in more of the statistical analysis and modeling end of things, which is actually really interesting personally for me, just from the standpoint of being interested in public health and epidemiology. And so I'm getting a little bit of a taste for that in a kind of circuitous way. I wasn't necessarily thinking that that's what I would be gaining, but it's a kind of roundabout way to getting to something I'm really interested in. So some personal takeaways just for me was a really deeper understanding and respect for how hard it is to be at the start of something scientifically. There currently isn't any methodology or standard for testing soil and looking for pathogens in produce. And so there is no rule book. The closest thing we have that's similar in terms of there being an established standard is actually for dairy, which is a really different system, but that's the closest thing we have to kind of a prior system of developing sampling to better control food safety risk. So kind of understanding better all of what comes with not having a guideline, not having just a set lab procedure, knowing exactly what you're doing, but instead problem solving as you go, figuring out new ways of thinking about things. And that was actually another thing, was how many different disciplines and different classes I ended up using a little bit of learning from. I have taken a lot of really random courses, and sometimes I'm like, where is this going to apply? How am I going to use this information? And it came back in weird ways that I wasn't expecting, but was really glad to have that knowledge. And so even if you don't necessarily see how you're going to use something, it will come back if you have that knowledge. It's never wasted. There's always a way of finding something, finding a way to use it. And the more you know, the more you're able to kind of translate between different fields, disciplines, and really get a better understanding for an overall project, rather than just being hyper-specific on just knowing one thing. And another thing I would just say is a general takeaway is that opportunities are what you make of them. So am I personally someone who's going to always stay in the field of food safety and applied food safety? Probably not. But I'm able to learn a lot still through this experience and find things that I am genuinely, truly passionate about, and able to further develop skills that I'll use for the rest of my life. So there's always opportunities for you to turn it into what you need it to be. And so figuring out a way to not just kind of experience something, but being really intentional about, okay, I want to learn this, I want to do this, here's how I want to grow, and thinking about that is really, really helpful. And finding balance, these programs are not easy. It is a large time commitment. Again, you will get out of it what you put into it, but it still is hard to sometimes balance all of that work and a personal life and taking care of yourself. And so you don't want to let one fall by the wayside, because if you do, nothing else is going to work. And if you want to try this, if you have the opportunity to, go for it. The worst that happens is they say no. The best thing that can happen is that it completely changes your life. I know this opportunity really did that for me, and I didn't expect that going into it. I didn't know what to expect. But be open to it, try new things, and always be willing to pivot when the time comes. There's some citations. I'd like to thank again the amazing Dr. Lloyd and Dr. Hind for all of the work they do in setting up a really, really great program and a really great opportunity. Specifically having the boot camp, I feel, is a really great way of making sure that you don't just kind of go into it blind, or not having an expectation for what standards you want to set, how to communicate. Even if you decide at the end of something like this that you don't necessarily want to go into scientific research, there's still skills you can learn that will be helpful the rest of your life. How to deal with interpersonal relationships in a workplace, how to do something new and be okay with failing and figuring it out, and knowing how to use your resources, going to the right people. Science is hard, but it can be really, really rewarding. And so if you get a chance and you want to do something like that, just pursue it. Again, the worst that can happen is you say no, and then you just move on. Any questions?
Go ahead and hand it.
A two-parter. One, what was the most challenging part of your experience, and what was the most exciting part of your experience?
Most challenging was adapting to a different field or discipline. I'm a person who tends to be a heavy control in the lab, having a lot more fine-tuned control of everything that goes on and knowing exactly where I can trace, okay, this is how it happened. In field work, it's a lot different. You don't control if it rains, if it's a good day to collect. If one of my fields, they planted over with soybeans, and so I just had to adapt to that and walk between soybeans like this. But adapting to the fact that my sphere of control is not infinite. I can't control the weather or the field, but I can still get valuable and relatively consistent data from it was interesting, as well as hard for me to adapt to that aspect of not everything is going to be perfect, but it's on the aggregate, zooming out and seeing the project from a lens a little bit less removed and less in the weeds of, oh, is this exact sample perfect? Versus, okay, I've taken quite a few. I have hundreds of plates. The data is going to be fine. And you don't know till you actually get in there and analyze it how valid it's going to be. The other thing was...
What was the most exciting?
Getting the chance to work on my own and get some getting the opportunity, my lab was really great about giving me the chance to work on my own and problem solve, figure it out from my own. And they really gave me the opportunity to control what I could in terms of, I wanted to do things specific ways. I wanted to make sure I sampled when I could get the E. coli as far as possible. I wanted to push it out 7 days so I could see the maximum relevant amount of die-off. Getting to actually have some say in what was happening and getting to contribute meaningfully. And now getting to write a paper is seeing something through all the way to the point where it's going to publish is incredible. That's something I genuinely only dreamed of or thought could only happen to graduate students. And so it's been just a huge opportunity to learn so much.
Were there any wrong turns in the research or anything that you stumbled upon that wasn't in the scope of the project that you thought, oh, this would be great to learn more about, but you had to put it aside to keep moving with what you were working on?
For me, that was definitely the APC counts. I went a little nuts trying to figure out like the validity and how to control it and how to properly set my ranges for enumeration. It just isn't something I immediately was able to pick up on, like, because I was defining my own rules. There aren't any rules for like standard plate counts in soil, which is incredibly microbially dense and diverse. And so trying to kind of figure out how to really rapidly learn everything I possibly could about microbiology specifically in terms of how plate dynamics even work. That was an infinite rabbit hole that I couldn't fully flesh out. Just there was no way I was going to be able to be the definitive end-all be-all on exactly what the rules should be for this. The other thing would probably be some of the more, at a certain point, okay, towards the very end of the data, actually, you can kind of see specifically with the drag swabs, we noticed that there was and have been consistently higher collection, or like it seems to recover total coliforms a little bit better with the BPW, but specifically for the drag swabs versus the booties. And that's a consistent trend, but it's, if you actually look at it, it's still within like a log of difference. Is that meaningful? It's kind of cool. It's interesting that there is a slight difference in how the drag swabs versus the boot swabs are kind of picking things up and able to support the microbial life that is on our samples. But at the end of the day, it's still within a 1 log difference. And then past 7 days, are you really able to get useful information to apply to food safety? Not really. You're not going to hold off on harvesting for 2-3 weeks while you dig down into the nitty-gritty details and store these samples. It's just, at a certain point, you have to let go with, yeah, this is kind of interesting. It's outside the scope.
All right, thank you so much, Ray. Go ahead and pass the remote on to Adam. Adam, you're up.
You guys hear me? Awesome. So my name is Adam Zwawi. I'm a student here at Parkland, and I'm going to talk to you guys about the friendliest bradyrhizobia. So this is going to be the research I did this summer with my grad student mentor, Amanda Griffin, and we did this research under the supervision of our lab PI, Professor Katie Heath, who is over at the University of Illinois in Urbana-Champaign, and she's currently the department chair of the plant biology department. This was an amazing opportunity that I had that I was very nervous about applying for. So right off the bat, just a show of hands, is anyone thinking about applying to RU this summer or in the future? One person? Okay, hopefully by the end of this talk, there's going to at least be 2 more people. So the research that I did was all about soybean symbiosis with a bacteria. The bacteria that we studied is what we call a nitrogen fixer. So the significance of nitrogen fixers comes down to nitrogen. The atmosphere that we have around us is 78% diatomic nitrogen, which means that we're going to find it in that N2 form. The thing about this N2 form is it's not very useful for life. Plants can't really make much out of it, and that's a problem because plants need nitrogen. They need nitrogen for their proteins, they need nitrogen for the genetic information, they need nitrogen. And they rely on bacteria in the soil to convert this nitrogen from the diatomic form into the NH3, NH4 form, which we call ammonia. The nitrogen cycle continues through nitrifying bacteria and eventually denitrification. And so bacteria are essential to the role of life. Something about this bacteria that we studied, although the nitrogen cycle kind of converts between its forms by bacteria, not all bacteria provide nitrogen the same. So this host symbiote compatibility is very specific. The plant that I studied is going to be the soybean plant, and its symbiote is going to be Bradyrhizobia japonicum. A lot of this specificity comes down to what actually happens in this friendship. So for this to happen is they need a place to be friends. That place is actually a nodule. And for a bacteria to nodulate into a plant, there has to be a chemical dance almost. They have to send the right signals, they have to get along, and eventually the plant will welcome the bacteria into its roots. So what does this look like? Over here on the left, we see a root nodule, and this is actually where we're going to find the bacteria. Over here on the other side, we have an example of a root that does not have any nodules. Right off the bat, you can see a pretty big difference. So this exchange is very specific. People are very different. Not all of us, even though we're all people, we don't get along with the same types of people. We have different personalities. The same way, or similarly, as humans have interacted with plants, we have bred slight differences throughout time. So if you guys might be familiar with dogs, think of like a wolf versus a Yorkshire Terrier. They're very different. The same way, even though they're both dogs. So the same way, we have different timestamps that we can mark the evolution of plants. We have wild plants. We have wild soybean, which if you actually look at this plant, it's very viney. It doesn't have a lot of fruitful yield, but it's good at growing in the wild, and it kind of does what it has to do. It can reproduce. Good for it. Humans got involved and they were like, okay, we want a bit more out of this plant. So they put plants that had similar higher yields of this wild type together, and eventually they got what we call land races, which is going to be a bit more of that agronomic traits, things that we can benefit from. We further this with more deliberate breeding practices, and we end up with what we call elite cultivars. And these are all the way on the end of the spectrum. So in my experiment, what we studied a land race and an elite cultivar, which the land race is 5th moon and the elite cultivar is 91B42. The reason why we chose this is we wanted to see what consequences there might be to the symbiosis as humans have involved themselves with this breeding pathway. So what we're really looking at is what's called a domestication syndrome. The idea here is that as we select for certain traits among plants, we might be inadvertently selecting, or without even realizing it, we're losing out on a lot of traits responsible for this symbiosis. So to put this into like something that might be a bit more common, if we know certain types of purebred dogs, while they look very pretty and they win a lot of awards, they don't live that long, or they have a lot of other health downsides. So we wanted to investigate that type of thing. On the other side, we have bacteria, so Bradyrhizobia japonicum. And the thing about bacteria is they're very fast to reproduce. And with that, they make a lot of mistakes and they mutate pretty quickly. So we wanted to choose 20 different strains to see if there were genetic differences, or if there are differences in these bacteria with how they go about this symbiosis. So we chose 20 different strains from the USDA Rhizobia gene bank based off of differences in age, geography, and frankly comprehension. So some of these bacteria we know for a fact that they're recommended by the USDA for inoculation. So the USDA actually tells farmers to put these bacteria on their plants with hopes of improving yield. Others have been sitting in the freezer since 1913, so we don't know much about them. They're very poorly understood. We don't have reference genomes for them, and we'd like to see if they're good quality partners. So our experimental questions are going to be, to what degree do our strains differ in partner quality, and are some plants going to be more susceptible to this nodulation than others? So to test this, we set up our experiment. We had to plant plants and we had to grow bacteria. On the plant sides, one of the things that was very important, since we're studying the effect of bacteria, is we needed to start with sterile soil. So what this meant was actually ordering soil from the greenhouse and sterilizing it. We have a machine over here called the autoclave, which I got to use this one. It's really cool. It uses temperature and pressure to reach high temperature and pressure to reach 121 degrees Celsius, which is really hot. I got a burn on my elbow. I left a scar. And so we sterilized our soil. We sterilized our containers as well, which are the pots that we're growing the plants in. And we put the soil in the containers. From here, we wanted to see how much nitrogen there is in the soil from the start. So we ordered these nitrogen testing kits and we tested how much nitrogen is in our soil. We got a baseline, we found that there's a lot of nitrogen. That's a bad thing, because if there is already nitrogen in the soil, the plant's gonna think to itself, why am I giving a lot of my valuable sugars, which I worked so hard to make, to this bacteria, when I can just get the same thing that the bacteria is giving me from a different form? So a big part of our experiment, took a couple weeks, was actually leaching the nitrogen from the soil, which just means I held a hose over the water for a couple hours and I washed the soil. From here, we were ready to plant. On the bacteria sides of things, I talked about how some of our strains have been lyophilized or frozen, desiccated, dried out, not alive, or not actively reproducing. And so a big challenge of this experiment was actually getting a lot of these bacteria back to life. The main goal from the different forms of how we got to them, or how they were received to us, is that we needed single colonies that we could grow a media from. The idea of having a single colony is that we don't want to start with two strains that might be slightly different. So we plated our bacteria and we put it in liquid media and we let it grow for a while. And from there we diluted our liquid media. We used this really fancy machine that shines light through the liquid sample, and it essentially tells us how much bacteria is in the liquid media. So we diluted them to have them all to a consistent amount, and this is called making our inoculum. So once we had our inoculum, we were ready to inoculate. We let our plants grow with their inoculum for a bit, and then we collected the data, and this is what we got. So This is data on the nodule count, which is after we harvested, we looked at how many nodules are in each plant. We looked at two genotypes. We have our 91B42 and our fifth moon, which are the different dots. We have our bacterial strain on the x-axis because we wanted to see the differences between the strains of the different treatments. And we have our nodule count on the y-axis. We found that the effects of the strain depends on the plant. However, there's not a significant, throughout the board, there was not a significant difference between the individual strains or the individual treatments. And this was a common trend that we found. For the above ground biomass, we found that a difference does exist between the two genotypes of plants. So the landrace and the elite cultivar grew differently, but we found that this was not a result of the bacteria itself. And we did the same for the below ground biomass. So we took the roots, we weighed them, and we did not find significant results here either. And so that leads us to our conclusion that under the specific conditions of our experiment, there's no significant levels of diversity and partner quality between the treatments under the conditions that we tested. And that's the caveat. So if we go back to our experimental setup, we had our plants pretty close together in the greenhouse. And since we're studying bacteria, there's always going to be this possibility of cross-contamination. We had these little container hats in between the plants to kind of mitigate that. We also hand watered to further reduce that impact, but inevitably, it's likely that some bacteria found its way into a place where it shouldn't be. And that would diminish the results, because if we don't have single strain treatments, then it's hard to say that there's a difference between single strains. Another thing that you might notice is that our containers are pretty small. I don't know how many people are from Illinois, have driven around the state, but if you've seen soybean on the side of the road, it's a pretty big crop. We have it growing in a very small condition. So if we let our experiment go on for too long and it actually outgrew the pot that it's in, the limiting factor isn't going to be the amount of nitrogen that the plant is getting. It's actually going to be the container that it's living in. And so under these circumstances, we have to ask, what if we grew them in more comfortable spaces? And that's something that we look forward to doing. My mentor, Amanda Griffin, is actually running an experiment which I'm helping her with, in which she's studying 50 different genotypes of plants. And she's using a mixed strain inoculation, and she's using these 1510 pots, which are about yay size. And I'm excited to see the results of that. So future works, we would like to confirm our results of the contamination idea. So we could do that by actually taking samples of the nodules, and we can do sequencing, which is a newer technology. It's been around for a while, but there are newer variants that are able to tell us essentially fingerprinting who is inside of these nodules. We'll be able to know who is who. So what was hard about this experiment? It was really hard to explore a new field. Before I took on this project, I had no idea about plants. I took like some general bio courses, but we didn't really dive too deeply into the plant world. It wasn't really something that I was interested in. And coming with that, there's a lot of new stuff to learn, and you don't even know where to, or at least I didn't even know where to start learning. And so the whole literature review process was a lot for me. It was very challenging. Another thing is that running an experiment is not like taking a class and a lab and that type of thing. Dr. Lloyd mentioned how a lot of research that we might do in the classroom environment might be canned. And that is a really good point. When you're running an experiment, I like to think about cars a lot. You could be a mechanic who is working on one small part of the car, or you could be with the design team and you're designing the entire car. And so you have to think about the engine, you have to think about what tires to put on, you have to think about what all-wheel drive, rear-wheel drive, all of that stuff. And that's something I found a lot this summer too. There was a lot of working with microbiology people, there was a lot of working with people in the greenhouse, timing things. Everyone does things differently, and so it's a lot to process. The other thing is having that balance of work-life culture. When I started this project, my family wanted me to be around a lot, but I also wanted to give a lot of time to this project, and so it was hard to balance the two of those things. What did I gain and learn from this experiment? I got to apply a lot of classroom skills in the real world. So when I did some canned experiments in lab, it was very cookie cutter. There are these skills that you use with these techniques, whereas in the lab, everyone does things differently. For example, with plating the bacteria, there are numerous different tools that you could use to actually take the bacteria from liquid media and put it onto solid media. And everyone has a different preference, and there are different norms and that type of thing. And I was very excited to see how it is that people go about choosing the one that they do. I also began to learn how to learn something new. So with that whole going into a new field, you kind of have to accept that you are a beginner. This is something that is way over your head, and you have to be ready to put in the work, and you have to be ready to experiment. You have to really want to take something out of this, and that was a very good takeaway for me. Another thing was clarity. So before this experience, I had really thought that I wanted to work in a healthcare environment. So I went about college with this image in my mind that I wanted to be a doctor, and I wanted to do this thing, and I did my CNA, and I did all of these healthcare related experiences. And a lot of that was from the fact that I hadn't really knocked on the door of research much. And so when I started to become willing to try a new experience, I found that in the process of studying things in a hypothesis-driven way, there is a lot of uncertainty, but with that is a lot of, it's very rewarding to go about finding something new. That being said, finding out that you don't like something is just as important as finding out that you do like something. And this is something that I learned from someone who I look up to quite a lot. And I found out that I didn't like working with plants, and that's a very important experience for me to take away. Is my desired career path going to be science in the future? I think 100%. I really liked working in the lab. I really liked working with new questions, but I don't think it's going to be very plant focused in the future, and that's okay. Part of my experience this summer was also outside of the lab. We had the opportunity with the MICRO-CCS cohort to volunteer at the science market, and it was amazing. We talked to lots of people, we showed them science things. We also got to go to the Chicago Field Museum. I got to see a giraffe's heart, and this is the Tabaski exhibit. They have an entire exhibit dedicated to Africa and African culture, and I thought that was the coolest thing. These are acknowledgements. I want to say thank you so much, Dr. Lloyd and Dr. Hind, for putting on such an amazing REU, for giving us so much guidance throughout the experience. It's really been fruitful. I don't think it would be the same without you all. References, any questions?
All right, we only have time for one question. No? You don't want to ask another one? You can ask that same question you were going to ask before. It's okay, I promise.
It's a two-parter. What was the most challenging part of your experience, and what was the most exciting?
Okay, the most challenging part of the experience was realizing that I knew nothing and that everyone did things differently. And even if I did something differently in the past, it meant absolutely nothing to me because I was in a new field and it is very important to learn. And so that was a very challenging thing for me to learn, and it was very hard. Something that I was very excited about was I could talk about like my lab experience and like everything that I learned in that sense, but it was amazing to make friends. It was really cool to see that other scientists are kind of just people, and I mean the friendships and the impact that I had from this summer, like outside of academics, it's invaluable to me. It was amazing.
All right, thank you so much, Adam. Really appreciate that. All right, well, folks, keep in mind the next of our Kaler Science lectures, unfortunately we won't have one next month because that is going to be right in the middle of all of our laser show rentals that we're going to have the first weekend of December. So we will resume them again in February. On the 7th of February, we will have Professor Sara Pedron Haba. She will be presenting about bioengineered disease models, I believe. Okay, so that's what we'll have then. Thank you all so much for joining us this evening. We're going to have Season of Light here very shortly. Let me bring up Carl, and I hope you all enjoy the rest of your evening and weekend.